Okay, I think we will start now. So, once again, hello to everybody, and thanks for joining us for the first episode of our new webinar series, the Discover Molecular Interaction series. My name is Vivian, and I'm your host today, and I'm responsible for scientific communication at Dynamic Biosensors. I'm not doing this alone. I am supported today by my colleague Stefanie Mark, who is a scientist in our team. So, hello also to you, Steffi. And in this webinar series, we will deal with a different kind of molecular interaction each month, and we will discuss the challenges of the specific molecular interactions, and we will also offer solutions and assays to characterize those interactions. And the focus of today's webinar will be bispecific antibodies. Bispecific antibodies are a hot topic. This, for example, becomes obvious if we are looking at the number of publications on PubMed, which have increased considerably in the recent years. So the focus of today's webinar will be emicizumab, which is a bispecific antibody, and it was actually the second that was approved by the FDA. It is used as a therapy to treat hemophilia A, but bispecific antibodies are actually used in many different disease entities, also in cancer or other autoimmune diseases, for example. Let's have a quick look at the mechanism of action of emicizumab. So usually upon the damage of a blood vessel, there is an intricate cascade of coagulation factors which get activated and finally result in the formation of a stable blood clot. One of these coagulation factors is factor 8, which is important to bridge factor 10 and factor 9 in order to allow for the activation of factor 10 by factor 9. Now, in patients with hemophilia A, factor 8 is missing, so thereby the development of the stable blood clot is disrupted. The bispecific antibody emicizumab can now mimic the function of factor 8 by binding to factor 9 and factor 10 and thus restoring the blood coagulation cascade. If we want to develop or also optimize a bispecific binder such as emicizumab that binds to two antigens on the surface of the same cell, we need to have specific tools in order to characterize all the interactions that take place in such a situation. In fact, for bispecific binders, the interactions are a bit more complex than for usual one-to-one interactions. As you can see in this scheme, you have to consider two different binary interactions, which then give rise to a ternary interaction, which is the stable complex here. So there is a different dynamic interplay of different interactions, which can be summarized in the term avidity. And in order to really characterize all these interactions, we need to fulfill certain requirements. First of all, the assay should have a high degree of biological similarity. So that means we need to be able to co-immobilize both antigens at the same time, as it is presented on the cell membrane. Also, the antigens need to be accessible for the antibody, and they have to have a certain degree of mobility. And finally, we also need to be able to control the antigen stoichiometry and not only immobilize them randomly on our biosensor. The second requirement is that we want to achieve a full kinetic information in our measurements. We need to measure the binding rates of both the binary interactions as well as the ternary interaction, and to measure the stability of this complex. And then from a practical point of view, we also want the assay to be able to be automated in order to achieve the throughput that is needed for the development of a bispecific antibody. The assay we are going to present today is based on the switchSENSE technology. With switchSENSE measurements, you can measure a plethora of biophysical parameters. And if you're interested in any of the other applications or parameters you see on this slide, you're of course welcome to contact us or visit our website. And you can also check out our future episodes of this webinar where we will discuss different applications of switchSENSE. But for now, I would just like to give you a brief overview of the basic principle of a switchSENSE experiment for those of you who might not have heard of dynamic biosensors before joining this webinar. So the foundation of a switchSENSE assay is the so-called helix chip. The helix chip has two measurement electrodes, and on these measurement spots, you can find DNA-based nanolevers. These nanolevers are labeled with a fluorescent dye, and you can functionalize it with any ligand of your interest. The analyte will then be in solution, and upon binding of the analyte to the ligand, the fluorescence will be quenched, or the fluorescent intensity will be changed, either increased or decreased. So the readout of a switchSENSE experiment will be this change in fluorescent intensity. Another fact I would like to mention here is that we have these two measurement spots, and on each spot we can have nanolevers with two different fluorochromes. So you can, in the end, measure up to 4 interactions simultaneously in the same experiment, or you can also do real-time referencing. But let's get back to our bispecific binders. As I said before, the situation for bispecific antibodies is a bit more complex than the usual one-to-one interaction. And we met this challenge by designing a specific nanolever that looks a bit different from the nanolever I presented here. So we call this nanolever the DNA Y structure. This DNA Y structure enables you to really closely simulate the conditions on the cellular membrane. Let me briefly walk you through the different components of this nanolever so that this point becomes a bit more clear. So we have this longer stem region here with the two arms of the Y structure that each contain a fluorescent dye, a red one and a green one. You can now hybridize your protein of interest, which is conjugated to the complementary DNA strand, to one of these arms. The Y structure has a very flexible linker region here, which allows the arms to move freely. So they have a high degree of rotational flexibility, they can move around, and they are very accessible for the antibody. So with this, you can fulfill the requirements for simulating the cellular conditions. You have both antigens immobilized at the same time. They are flexible and accessible. And what is very important is that you create a specific local concentration of both binding partners here. And they are in a defined distance and a defined stoichiometry. You can now use this Y structure to either measure the binary interactions if you modify only one arm. In our example, this would be having only factor 9 functionalized to the Y structure or only factor 10. Or you can measure the ternary complex by functionalizing both factors at the same time. The workflow for such an assay is very straightforward. You basically just mix all the components, and then the functionalization on the chip surface will be performed completely automated in the biosensor instrument. So once you have your surface functionalized with your antigens on the Y structure, you can perform the kinetic analysis. So let's get to the data part of this presentation. At first, we are going to look at the binary affinities for each of the factors. You can see here the Y structure modified with only factor 9 in green or modified with only factor 10 in red. In these kinetic traces, you see first the baseline of the fluorescence, and upon binding of the antibody emicizumab, the fluorescence is decreased. The higher the concentration of emicizumab, the stronger this decrease in fluorescence will be. When we then flow buffer over the sensor surface, the emicizumab antibody will dissociate again from its binding partner, and this causes the fluorescence to recover to the baseline again. Now, if we look at this interaction, we see already a very interesting result about the binding mode of emicizumab. That is that the two different arms of emicizumab have actually a very different binding affinity to their specific target. We see that the dissociation of factor 9 is very fast, which results in a KD in the micromolar range. On the other hand, the dissociation of factor 10 is more slowly, and this interaction is more stable, resulting in a KD in the nanomolar range. So now what happens if we immobilize both targets at the same time and look at this cooperative binding or the ternary complex? You can see here again in green the binding kinetics for factor 9 and in red the binding kinetics for factor 10. But this time we have both antigens on the Y structure. You can see that in presence of factor 9, the binding kinetics of factor 10 do not significantly change. In fact, they stay almost exactly the same as if you are looking at factor 10 alone. In contrast to that, the binding kinetics of factor 9 drastically change if we have factor 10 present. You see that we don't have a simple one-to-one binding anymore, but we see a biphasic dissociation now with a faster component, which is very similar to the binding of factor 9 alone, and an additional slower dissociation, which is actually very similar to the dissociation of factor 10. So from these data, we can conclude that the presence of factor 10 stabilizes the binding of factor 9 and emicizumab. So what can we conclude from these kinetic data with respect to the binding mode of the antibody? First of all, we can say that the association is probably mediated by factor 10 as it has the faster on rate. And if we have factor 9 in proximity due to the Y structure, it will lead to a fast handoff to factor 9, and this ternary complex will be formed. When we look at the dissociation, we saw that the ternary complex is mainly stabilized by the factor 10 binding, which was the more stable interactant. It is very likely that there will be a release and a rebinding of factor 9 and emicizumab in a dynamic range because factor 9 was the less stable interactant. However, due to the stable binding of factor 10, this ternary complex will be stable until the point where factor 10 is also dissociating. So why did I point out here that this is happening at low concentrations of the antibody? For this, we can have another look at the dissociation of factor 9. As I said before, this is now a biphasic dissociation, so we see this fast component first, and then we see a slower component in the second part. Interestingly, we see a difference for the different concentrations of emicizumab, which is not very usual for dissociation kinetics. So the biphasic dissociation can be described by a bi-exponential function, which you can see here in light blue. This bi-exponential function consists of two mono-exponential functions, one for the faster dissociation in black and one for the slower dissociation also in black. Using this, we can derive the contribution of the fast and the slow dissociation. And if we now look at this for increasing concentrations of emicizumab, you can see that the fast dissociation amplitude increases, whereas the slow dissociation amplitude decreases. So this has very important implications for the dose response of the bispecific antibody. From this, we can conclude that once we have a very high concentration of the bispecific antibody, we see something that is very similar to the so-called hook effect, where the antibody binds with the binary affinities with one antibody arm binding to each of the two antigens instead of the formation of this ternary complex. So if we increase the concentrations of emicizumab until a too high concentration, this will decrease the efficacy of the antibody because there will be no more ternary complexes formed, and instead we will have this effect. And why should we care to characterize these kinetics in such great detail? Emicizumab is a very nice example to demonstrate the importance of kinetic rates for the in vivo mechanisms of therapeutic antibodies. If we look at the in vivo situation, usually in a healthy blood vessel, we will have the coagulation factors in solution. So this is a binding situation which corresponds to the binary affinities where we have each arm bound to a coagulation factor, but the half-life of this ternary complex will only be about 1 second due to the very low affinity of factor 9. This is not enough for the catalytic activity to take place and for factor 10 to be activated. But once the blood vessel is damaged and we now have the two factors presented on the surface of the platelets, we have this fast handover that I described before, and we have the stabilization of this ternary complex due to the kinetics of factor 10. Now the half-life of this ternary complex will be about 20 seconds, and since the catalytic half-time is about 15 seconds, this is now stable enough to allow for the catalytic activation of the coagulation factor and thus for the therapeutic effect of emicizumab. So if we summarize this assay format briefly, we have shown that you can analyze bispecific antibodies with the DNA Y structure because you can analyze the ternary and binary binding modes at the same time and differentiate between them. Also, the Y structure offers a very high degree of biological similarity due to the controlled local concentration and stoichiometry and also the co-immobilization of both targets at the same time. The assay format is very versatile. We measured a very high range of different KDs with this setup, and it's a very straightforward assay. And you can also scale it up to make it suitable for antibody screenings. Now I would like to just give you a very quick outlook, because if you are interested in bispecific antibodies, you might also be interested in antibodies that bind to two targets on, for example, cancer cells. And in those cases, it might be interesting for you to look at the binding mechanism on the cells directly. And for this, you could use the real-time interaction cytometry, which allows you to measure the real-time binding kinetics directly on living cells. If you are interested to find out more about RTIC, you can also visit our website or check out our YouTube channel, where we have some more content on RTIC, and there will also be future webinar episodes about this application. But for now, this has been everything I wanted to talk about in this episode today. I hope I could give you a good overview of how to use switchSENSE and the Y structure to measure your specific bispecific binders. And thank you very much for your attention, and we are happy to answer your questions now. Okay. So I will quickly check as I said before, you can either use the chat or raise your hand or use the Q&A, as some people have done. So let's just start with the first question I have here. The Y structure construct. Someone is interested in how big the distance of the two antigens is, or how big the Y structure is in total. Steffi, maybe you can elaborate on that.
Yeah, thank you for the question, Vivian. Can you please go back to the slide where you showed the Y structure in detail?
Sure.
So that everyone knows what exactly we are talking about? Yeah, this one. Okay. As it's noted here, the total distance of the two antigens that are immobilized on the Y structure can vary a lot. We determined this distance to be between 0 and 40 nanometers. This depends, of course, on the size of the two proteins. So the 40 nanometers, that would be about the length of the two arms together, plus a little bit extra.
Thank you. I would like to point out that this is also one of the big advantages of the Y structure, that we have this mobility and the different distances, because this is actually what is happening on the cell too, that you have antigens that are mobile in the cell membrane. So this can be very important to simulate what's happening in vivo. Okay, let's get on with the second question. Is the flow rate of the device high enough to prevent mass transport limitation and rebinding, especially for fast interactions?
As we have seen here in this example, we can resolve quite fast interactions with the Helix biosensor. In this case, the interaction between the antibody and factor 9 was very fast. So the dissociation happens in a few seconds. Yeah, as you can see here on the upper left. And especially for those fast interactions, it's important to measure at high enough flow rates so that you do not measure a far or a wrong association rate that might happen due to not enough antibody delivered to the surface in time, or a false off rate that happens due to rebinding. If antibody that is detached from the surface isn't washed away fast enough and can bind again at a different place. We have taken care with the Helix biosensor to offer fast enough flow rates so that those two cases can be excluded. And even here, with those very fast interactions we have seen here, we do not see any measurement artifacts that would hint at rebinding or mass transfer limitation. So we offer a maximum flow rate of 200 microliters per minute, For those that are of you that are used to measurements on SPR, there you normally have even higher flow rates, but also a bigger diameter of the flow channel, so that even our seemingly lower flow rates lead to very efficient replacement of the fluid.
Mhm. Yeah, I think this is also very important with respect to bispecific antibodies, because also in the case of emicizumab, for example, you don't want the antibody to be stuck on the antigens for an endless amount of time. You usually aim for rather fast binders, so it's important to accommodate that in your assays. Thank you. Now that we are talking about time anyways, maybe I choose the next question that sort of fits to the topic. How long did the instrument run for one experiment, and how long was the corresponding hands-on time?
Yeah, so our device offers a high rate of automation. This means that the hands-on time is relatively low. So you basically need to prepare your samples, create your dilution series, put it into the instrument, and then start. The rest is handled by the instrument itself. This leads to a very low hands-on time. For a typical experiment, where like the one where the data is shown here, this would be perhaps half an hour, depending on how fast you are in the lab. The runtime of the instrument is a bit harder to define, because this depends a lot on the interaction you want to examine. The interaction we have seen here is very fast. So if you look at the x-axis of the plots, you see that one run for one concentration takes less than 5 minutes. So the whole experiment, as shown here with those 4 plots, can be done in a few hours. If you're looking at a more stable interaction, as it is typical with bispecific antibodies, I have to say, we are looking at dissociation times that are rather in the range of hours than minutes. We can measure with the device up to a few hours, like 10 hours at a time. But of course, if you want to measure 5 or 6 different concentrations with 10 hours dissociation time each, we are looking more at an over weekend experiment than a few hours. But luckily, the device handles it all by itself. You just turn it on and let it run, even over the weekend.
True. Okay. Now maybe some more technical questions. Somebody asks whether the attachment of the antigen to the Y structure would impact the affinity and the kinetics. Maybe we go back here for this.
That's a very good question. As you can see here, our proteins of interest are conjugated to the corresponding DNA strands that are complementary to the DNA strands on the Y structure backbone. We have 2 different DNA constructs, the ones that are displayed here in blue and red, which have a different sequence. Like this, we can make sure that the proteins of interest end up attached to the right arm of the Y structure and thus to the right fluorophore. The DNA strands are connected to the proteins of interest with a chemical reaction. We use amine coupling for this. And this is a random process. This means that every DNA strand is attached to its protein in some random place. Of course, it might happen that a DNA strand attaches to the protein of interest exactly at the position where the antibody would bind. But as this is a random process, we can basically guarantee that we will always have the biggest population of antigens that are able to bind to the antibody without any disturbance.
Okay, I would say I will choose one last question before wrapping up the session. Let's see. Yeah, maybe something about more applications of the Y structure. Can we only measure bispecific antibodies, or will we also see avidity effects when measuring a monospecific antibody?
Yeah, as you are completely free in choosing which proteins you want to couple to the Y structure, you can, of course, couple the same protein twice and thus measure a monospecific antibody. There you will see the same kinetics on if you look at only the green or only the red arm, as in our control experiments that were shown for the bispecific antibody. But if you put up your protein on both arms of the Y structure, you will see a decidedly different kinetics, as you then see the avidity effect. Normally, in those cases for a monospecific antibody, you will only have one on rate because it's the same for both arms. But you will see a clear distinction between two different off rates. Again, the faster one is the affinity case, where only one arm of the antibody is bound. And then the avidity case, this is the slower off rate, where both arms of the antibodies are bound. As both control experiments with both colors result in the same rates, normally we do only one of them. And thus, experiments with monospecific antibodies are even easier and faster than those with bispecific antibodies.
So if you have an antibody you would like to test, please reach out to us. We are always happy to organize a demonstration in your lab with your specific molecules, or you can also send us your molecules and we will test how it looks like with our system. So I think if there are no more questions, I think I will wrap up this webinar now. And thank you very much, Steffi, for supporting me in this first episode. And thank you all very much for joining. I hope that this gave you some ideas for your investigations in bispecific binders. Please reach out to us, tell us what you thought, tell us any other questions you might have. You can send me an email, you can go to our website and use the contact formula there. And I hope to see many of you in our future episodes of this webinar, and have a great rest of your day and see you soon.